What kind of research are you working on?
In our laboratory, we are developing technology to study proteins. And these technologies help basic biologists and also clinical researchers to better understand the biological and medical mechanisms of diseases and physiological processes. And in particular, we're interested in what is the footprint of genomic alterations, mutations, for example, on signaling pathways in cells, and if we can identify new therapeutic targets and biomarkers using our technology. What we do is analyze proteins, thousands of them, also their modification states, how they're turned on and off. And also those new machine learning tools will help us to predict, hopefully in the future, how drugs work in cancer and also how the functional state of a human heart is.
How do we understand the mechanisms of cancer development by studying the proteins?
So by reading our proteins, we can study the consequences of mutations. For example, proteins encode for growth factor receptors or for metabolic enzymes, and when those change, signals are altered in cancer cells that make the cells divide or go to a different part of the body when cancer cells metastasize. And therefore it's very interesting to study the proteins, which are in the end also most of the drug targets. So if you have one of those new chemotherapeutic drugs or targeted drugs, they always have a protein that's the target, and it's very important to read out the quantities of those proteins in order to figure out which patients may benefit or not from a certain treatment.
And you have studies about colon cancer too, right?
We analyze lots of different tumor types. With collaborators in Berlin, we do a lot of colon cancer research because they have a large-scale library of xenograft models. Those are human tumors that they can grow in mice in order to study individual drugs. So in the clinics, patients often see lots of different drugs, and it's hard to understand what drug was actually helping the patient. And when you isolate the drug-tumor combinations in a mouse model experiment, you can better understand which drug is actually effective, and hopefully identify also markers that will allow you to predict in a patient later on if a drug will work or not.
If you find an effective drug when performing research on mice, can we be sure that this drug will help people, human patients too? So
basically we help generate hypotheses and give answers of what drug effects correlate to a certain protein level or biomarker that we can measure, and then we can make recommendations to clinicians. But in the end, clinicians have to define in clinical trials to see if the new mechanisms that we help to understand can be used also in the clinic in the long run, and what is also feasible to transfer from the basic research biology lab into the clinic.